All right, ladies and gentlemen, we are beginning our mutations activity group work. Now that I'm making this video, I don't know how I'm going to be doing the group choosing, but hopefully by the time we get to class, I'll have that figured out. Those of you who would like me to read through this entire thing, I will do so after I go through all the directions. So stick around if you want me to read this to you. But first, directions. First thing you're going to do is read through this first page. I know you don't want to, do it anyway. And then you are going to answer these questions, the pre-lab questions. That is stuff that you would already know based on your notes. So if you don't know it, talk to your group members or look in your notes for it. When you get to number 5, this is a codon chart. I typically show you this round version here. The keystones, I think, like to use this square version, so I want you guys to have a chance to see this and use that. How this works is you get 3 letters to look for in the codon, G, A, U right here. The first letter is going to come from the side. The second letter is going to come from the top. So your first letter is G. So you come over here and look for the G. And then your second letter is A. So you come up here and look for the A. So where this column and this row meet right here, you're going to look for GAU, which is ASP, which is short for aspartic acid. If you prefer to use the round one, I will have this posted for you. And how this works, if you still don't remember, GAU, you start in the middle of the chart and then you work your way out. So GAU, and that is aspartic acid. You see how that works? Either one of these codon charts is fine to use. Now, procedures. Using the DNA strand on the data sheet, which is right here, you are going to transcribe the DNA into mRNA. So here is the DNA that I gave you. You are going to turn this into mRNA using your base pair rules. I'll do the first couple as an example. T pairs with A pairs with U because RNA doesn't have T's, it has U's. C pairs with G. And then you're going to keep doing that all the way down to make the correct mRNA sequence. And then you are going to use your codon chart here or the round one to figure out what it stands for. So AUG is methionine, and you would just put that here. And then keep going all the way down. And then we are going to mutate this stuff. So we are going to mutate this DNA, change it somehow, and that's what we're going to write. We're going to write the changed DNA right here. How are you going to change it? You are going to randomly select how that works. So the first nucleotide that we will mutate is going to be the 4th nucleotide in that DNA strand. 1, 2, 3, 4, right? This G is going to get mutated somehow. How are we going to do it? With this chart, I gave you numbers. You are going to use this random number generator to select the action. So you come here, and you only need this part right up top. So lower limit is 1, upper limit is 6, because we're only going to have 6 options, and you hit generate, and I got number 2. Make sure that no one else in your group has the same number as you. If someone else has the same number you got, one of you has to just hit generate again. Make sure everyone in your group has a different number. I got a 2, so I'm going to substitute with a C. So I'm going to rewrite the DNA, but on the 4th one, instead of a G, I'm going to put a C. I had a 2 there. So rewrite the DNA, but with my mutation. And then I write the RNA with the mutation. And then I put the amino acids with the mutation. Notice With the mutated DNA, I gave you an extra space here. That's because some of you may get an insertion. You add something in. If you did get an insert, if you got number 6, generate again 1 to 4 to figure out if you're getting an A, C, G, or T. I say that again. If you randomly generated a 6, you are going to insert the nucleotide. What are you inserting? Go to your random generator, hit generate. I got a 4. So I get number 4, I'm going to insert the T into my DNA. If you get that insertion, you are going to put it after the G. So you're still going to write TACG and then your new number and then continue with it. So you'll have an extra space to fill. And then put your sequences here. Trial number 2 is going to be exactly the same way. Go back to generation, random generator, pick another number, and this time you are going to mess with the 6th nucleotide. So again, correct DNA, correct RNA, correct amino acids, your randomly generated number, and then your mutated DNA, your mutated RNA, and your mutated amino acids, and then answer the questions. You can work with your group members for these questions. Since your group members have all different numbers, you'll have different sequences. That is entirely your own, but you can work together for the questions. All right? All right. Now, for those of you who want me to read through this, I'm going to do so. If you want to do this on your own, read it by yourself, then good luck. I will see you in class. Mutations activity. Introduction. DNA is a genetic material made of nucleotides. Last unit, we saw how proteins were created through transcription, DNA to mRNA, and translation, mRNA to linked amino acids. However, in this unit, we want to see how those processes can go wrong and create mutations. In this activity, you will investigate a gene mutation in which there is a change in one nucleotide. This can be a point mutation called a substitution, where a base pair is swapped with the incorrect base, or it can be an insertion or deletion, in which cause a frame shift mutation. You will see how these changes affect the way the message transcribed to RNA and translate to protein. Explore the effects of gene mutations. Concepts. By the end of this lab, you should be refreshed on transcription and translation, understand how a point mutation can alter a gene, and understand the different types of point mutations. Background. DNA is an example of a complex biological polymer called a nucleic acid, which is made up of small subunits called nucleotides. There are 4 possible nitrogen bases in DNA: adenine, guanine, cytosine, or thymine. The nitrogen bases will preferentially bond with only one other nitrogenous base: adenine with thymine, guanine with cytosine. The bonded nitrogen bases are called base pair. This DNA is used to complete protein synthesis. During protein synthesis at the ribosome, mRNA sequences are read and translated into amino acids. Another area of the tRNA transports a specific amino acid. The amino acids are linked together into chains by enzymes to form proteins. Depending on the DNA sequence, the 20 amino acids are brought to the ribosomes by transfer RNA, tRNA. An infinite variety of proteins can be formed from the 20 amino acids, which can occur in any number and in any order. These amino acids are specified by codons, or 3 nucleotides in a row on the mRNA. If the mRNA is copied incorrectly during transcription, the mutation will occur. A gene mutation is a change in a single base pair of a gene. Point mutations, or single nucleotide polymorphisms, involve only one nitrogen base change of the 3 nitrogen bases in a codon. Perform this activity and witness the change in a single gene mutation in the DNA can have on a resulting protein. Pre-lab questions. This lab shows the effect of point mutations, which will cause gene mutations. Only affects one gene. What is the difference between this and a chromosomal mutation? 2. Describe each of these mutations. Substitution, insertion, deletion. Number 3, write the correct type of mutation that occurred in creating the mRNA strand for each of the following examples. So the original DNA, ATC, CGC, TTACG. And here we have mutated DNA or mutated RNA. This is incorrect. All right, so mutated RNA. And you have to tell me what kind of mutation it is. Is it a substitution, insertion, or deletion? Number 4, an insertion and a deletion will cause a frame shift. Explain what this means. Is this more or less likely to cause a noticeable mutation than a substitution? Number 5, I already explained. Refresh your knowledge on how to use the universal codon chart below. So AUG codes for what? CCG codes for what? GAU codes for what? UAA codes for what? Procedures. Using the DNA strand on the data worksheet, transcribe the DNA into mRNA. 2. Using the universal codon chart found in the pre-lab, translate the mRNA into the corresponding amino acid sequence. 3. The first nucleotide that will be mutated is the 4th nucleotide in the strand of DNA. That is this one. When you rewrite the DNA here, you are going to mutate this 4th nucleotide, this G. 4, use a random number generator to select your action. These are your actions down here. No student in a group may use the same number. So if someone else in your group got 3, then generate another number if you also got 3. So number 1, if you get a 1, you are going to substitute that nucleotide with an A. So you'll get rid of this G and you'll put an A instead. If you generate a 2, you're going to substitute it with a C. If you generate a 3, substitute with a G. If you get a 4, substitute with a T. If you get a 5, delete the nucleotide altogether. So when you put it in, TAC, TAC, and then delete. Do not put the G. Go right to this T, but do not leave a space. Just move the T over. And everything else moves over with it. And if you get a 6, you are going to insert a nucleotide immediately after that specified nucleotide. So if you got a 6, you're going to leave this G alone and you're going to insert something afterwards. So TACG, TACG, and then whatever letter you are going to put in there, and then this T gets moved over, and everything else gets moved over as well. What letter are you putting here? You are going to go back to your random number generator, set it up for 1 to 4, hit generate, and this time I got a 4 again. So 4, I will put a T. and then continue making it, and make your RNA, make your amino acids. Write the complete DNA sequence with one mutation, I said that. Complete the mRNA sequence, translate the amino acid sequence from the mRNA, and then circle any differences from the original protein produced. So here's your original protein you're going to make, and here's your mutated protein. It says circle, you can highlight up here. Highlight any amino acids that are different between the two. And then you are going to repeat this activity again down here, trial 2. This time, you're going to mess with the 6th nucleotide. So this one here. Same rules though, just with this nucleotide. And then you're going to answer the questions at the end. Numbers 1 to 6. And that is it, ladies and gentlemen. Have good luck on this. Have a nice day. Ask me questions if you have them. Good luck.